Okay, boys. So you know that first of all, to clarify you that you have 5 topics coming in your exam, and these 5 topics are 1.1, 1.2, 1.3, 2.1, and 2.2. Okay. So 1.1 is about we will go from these one by one. So this is we will start from 1.1.1. So where it says that you have to talk about the van der Waals forces, covalent bonds, and ionic bonds. So I will try to brief it very simple. Remember only one word which is very important between them. We will go over one by one. So first thing that you have to remember only one word about these, which will help you out to answer them in the exam, is that, wait, let me take the red. Okay, so first of all, ionic bond. Whenever you see ionic bond, it means that transfer of electrons. Okay, so whenever you see ionic, it means transfer of electrons. So this is the hint, like how you can recognize what type of the bond we are talking about. And this is the strongest bond, okay, out of the 3. Then we have the covalent, which is the sharing of electrons. Okay, we have sharing of electrons. Have now here the covalent bond, which means the sharing of electrons. So remember, like whenever we talk about the covalent bonds, it means sharing of electron. This should be very clear to you. And the sharing of electron, one of the best example we have studied is water molecule, like the sharing of electron between the hydrogen and oxygen. Then we studied about the van der Waals forces, and these forces are the forces which are normally between water also, like between water and water molecules, just like you can say just like hydrogen bonding. We don't have here hydrogen bonding. We can write here as an extra. I can write here hydrogen bond. So if we say hydrogen bond, means that it is between 2 water molecules. Let us move to next. We have to discuss the examples of the van der Waals forces in biology. So remember, we studied one of the animals who have it on the foot. There were small spikes, and these spikes were like help the animal to walk over the walls, or so to climb over the wall in the lizard, let's say. So these are kind of the van der Waals forces which are very weak. After that, we have chemical reaction. You study very well in chemistry that you know that if we have A plus A plus B, it's very easy. A plus B gives you AB, where A plus B are the reactants and AB are the products. We have studied about the enzymes. Enzymes play a very important role in controlling the reaction. Like the enzymes are proteins, and they speed up the rate of the reaction without being used in it. After that, we discussed already about reactants. We discussed about the products. You can say from here, before arrow you have reactants, after arrow you have the product. Whatever is the reaction, it does not matter. It doesn't change the products. Activation energy. Yes, any student who just give me a brief about activation energy. What do we mean by activation energy? Yes, Abdullah Said.
Is the minimum amount of
It's the amount of energy.
Yes, very good. Minimum amount of the energy needed for the reaction to start, or you can say that needed for the reactants to convert into products. So it is the minimum amount of energy. And you can decrease this energy by using what?
Enzyme.
Yes, very good. Enzymes in industry and in we call them as a catalyst, and if these catalysts are in our body, we call them as the enzymes, and all enzymes are proteins. Remember, the catalyst or the enzymes just decrease the activation energy, they just speeds up the reaction. They don't increase the amount of the products. That's very important to know. In this diagram, you can see you may be asked to label like what is x plus y and what is xy. Yes.
X plus Y is the reactants and XY is the product.
Very good. And you can see here the two lines. One line is going up. That means that it has a high activation energy. And the second line is below. That means that it has a low activation energy. And this key is there that tells you if you have a full dark green line, it means that without enzyme. And if you have a light green line, it means that it is with enzyme. So the next question where it says that describe the functions of enzymes. We discussed all enzymes. Boys, remember, enzymes are made up of what? Enzymes are made up of
Protein.
proteins. Very good.
Proteins.
What do they do? They speed up the rate of reaction, and they
Chemical
do not,
reactions.
yeah, they do not increase the amount of the product, and they are never used up in the reaction. So you can take them back if you want. This model, which has all these three pictures, is called what? What is the name of this model?
Like a lock and key.
Yes, lock and key. Very good. So the name of the model is lock and key model. So what is the meaning of lock and key? I will go over from this picture. You can see over here, this blue down is an enzyme. Okay, this is an enzyme, and the hole on its surface, like this area, is
Lock, mister.
this is called as what?
Activate site.
Active site. And active site is a place where a specific molecule will bind, and that is called as a reactant. Sometime we say it as a reactant. Okay, we don't say it as a substrate. So remember, in the exam, maybe they say you this is a reactant or this is the substrate. So don't get confused. Okay, so this is also known as reactant. So this reactant will bind with the active site on the enzyme, and each substrate is very specific. Each enzyme is very specific, like the locks in your home. Okay. Then,
Mister, you can't say it again because I don't understand.
okay. Rather, I say, let me choose some student to say. Anyone else, please, can help Faris to understand lock and key?
Mister, the enzyme is like a lock,
Okay. Yes.
but the substrate is the key.
Okay. Very good.
And when the key and to unlock the enzyme, it's called
Enzyme.
enzyme separate complex, and then
Yeah.
it produce products.
Yes, very good. So you can see here, Faris, in this picture, you can see you have three enzymes. You have enzyme type 1, you have enzyme type 2, and you have, wait, it's not working. Okay, so you have enzyme type 1, you have enzyme type 2, and you have here enzyme type 3. Each enzyme is binding with a different substrate. Yani why different substrate? You see here is a triangle, here is a hexagon, and in the first there are the ovals. Why? Because of what?
The different
The difference.
active site of the different active site.
Yes, the difference is in their active sites. Very good. So each enzyme is very specific to its substrate shape. If I replace the substrate to go, yani substrate 2, and if I say that this substrate go and bind here, will it be able to do any enzyme substrate complex?
No.
No. If I say that one substrate 1 to go and bind with enzyme 2, will it make any reaction? No. So
No,
that's,
mister.
yes, these are not possible. Yani these are not possible. The reason is that they have a specific active site and that only binds to a specific substrate. Good. Let us move next. we need to see the reaction is endothermic or exothermic. This is very easy to identify. Just look at the reactants energy and look at the product energy. Okay.
Mr. If the reactance is low, it's endothermic.
Very good. And if the product is high,
It's exothermic.
no, you didn't listen to my statement. You said if the reactant
If the
energy is
reactance is high, it's exothermic, and if the reactants are low, it's endothermic.
yes, very good. But you can also say the same on the basis of the product. Okay, now let's talk on the basis of the product. If the product has high, then it is
High.
what?
Endo.
Endothermic.
Yes, and if the products
And
have
it's low, exothermic.
yes, very good. So you can say it on the basis of the product as well as on the basis of the reactants. Okay. Now the factors which affect the enzyme activity are temperature, pH, enzyme concentration, and substrate concentration. Normally, the enzymes which work in our body are having a temperature of 37. If you change the temperature and go above like 38, 39, and 40, what will happen to them? They will denature.
They
The
will have
enzymes,
aging and disease like this.
yeah, enzymes will stop working. Enzymes will stop working because the enzymes will be denatured. That's what we have to say. Reaction will be stopped. Okay, let us go now to next topic where it says the identify solubility. How we define solubility?
Mister, it's like the
The ability
Like
of the
Mr.
undissolved.
Sudar,
Undissolved.
One of you said that it is the ability of the solute to be dissolved in solvent. Yes, like let's say you take sugar and you take water, you dissolve them. So how much
yes,
sugar
Mr.
you can dissolve? Okay, how much sugar you can dissolve is called as
As much
solubility.
as you want.
Yes, but there will be a
much
some
as it's
level after that you cannot dissolve. Okay, and no more sugar can be dissolved. So means that the solubility of the water is finished. That was the maximum solubility. Now from here to we have to know very important thing, that is some of the solutions are heterogeneous and the other solutions are homogeneous. How we distinguish them?
Homogeneous.
Mr.
Mr. The
Heterogeneous
homogeneous is evenly mixed, and heterogeneous is not evenly mixed.
Yes, very good. So here we have mixtures or the solutions. Homo means same. Homo means same. Like evenly mixed, for example, coffee. And then hetero is like a mixture which is not evenly mixed and it has distinct particles shown easily. For example, biryani, for example,
Mister,
the salad.
Seawater.
Yes.
Mister,
Seawater.
in the homogeneous only one phase. The heterogeneous, if you see two or more phases, like
Yes.
oil and
Yes.
water, you see two phases.
Yes, yes. So there are now heterogeneous is classified into two more. Either it is a suspension or it is a colloid. So make sure
Like
you are able to distinguish between them. Suspension is like when you mix them and after some time you leave them and after some time everyone is like stop.
Like water
They
and
have
oil, Mr.
yes, yes, very good. Like water
Like a
and
barrier
oil.
between them.
Yes,
Like a
sand and
barrier.
water. Bravo, bravo. But in collide, they keep moving. They keep moving. Yani they don't settle down. You can see them all the way moving, moving, moving. Like fog, ratuba. So when you have that, you see it's like always moving in the air. Okay, fine. Let us go back and see. This is a water molecule. It is made up of hydrogen and oxygen. It is called as polar. Why we call it as polar? Why we call it as polar?
because it has a bent
because
Mr.
spin
it
on
Because
it
have
it's
and the
positive.
center or horizontally charged balls.
Yes, because there is an there is an unequal charge on water molecules, slightly positive and slightly negative. So if you see in this picture here, in this picture, you can see that
We have
hydrogen,
2 positive and 1 negative.
yes, yes,
Yes.
uneven charges. Okay, and if you see here in this picture, this is a water molecule, this is a water molecule, and let's take the example. This is a water molecule. All water molecules are linked to each other with these dotted lines, and these,
Mister,
yes,
when the water molecule become an ion, it can be not polar, right, Mister?
yes,
Like an
yes,
ion 2O and sharing the electrons of
yes,
each.
yes, yes. But here, for the time being, if you see at these two water molecules, the oxygen of one water molecule is binding with the hydrogen of another water molecule, and they are resulting in the formation of what? Hydrogen bond.
Hydrogen bond.
Yes, and that is shown as a dotted line between them. Okay, and you must remember that the maximum number of hydrogen bonds
4.
you see is
4.
4. 1, 2, 3, 4. So this is 2, this is 3, this is, sorry, 4. So these are the 4 you can see over here also. Okay, so 4 hydrogen bonds. Now water has either adhesive property or cohesive property. So we need to know like what type of property it is, adhesive or cohesive. Okay, so here we have adhesive and cohesive. So remember, cohesive when it stick to another water molecule. Remember, cohesion, water to water. So you see over here, yani this is one water molecule and this is
Mr.
another water molecule. Yes, I give you another example. Look, this is let's say a beaker. Okay, and you have all water molecules here, yani this is all water molecule. Now what you know that if this is a water molecule and this is a water molecule, and if they are connected with one another, let me make them little far. So if there is another water molecule here, sorry, 1 minute, let me try to make it like this. Okay, now it's easy. Now you see 3 water molecules. Mainly there are hundreds of the water molecules here, millions maybe, but you see only 3 water molecules. Now I will change the color to make it easy. Let's say this water molecule is binding with the beaker wall, or maybe this water molecule is in the plant and binding with the plant stem or the plant root. So now water with the wall, water with any other surface. So such force is called what? When the water is binding with a different surface, what it is called? Adhesion or adhesive force. Okay,
Adhesive, Mister.
yeah, adhesive. So if there is a bonding between the two water molecules, like you see these two water molecules, if they bind together, then this is a cohesive force. Okay, remember they bind together, so we will call them as this binding, like a hydrogen bond. Okay, so such type of bonding will be called as cohesive. I will write it here. Water is the best solvent. Water is a polar molecule. We studied it already. There is a slight positive and slight negative charge. We studied already. Different types of solutions we discussed. These are like more examples: solution, suspension, and colloid. Remember that suspension and colloid, they are heterogeneous, while solution is a homogeneous. And what is same between them? They are all mixtures. pH, we studied about the pH scale. How many values are in the scale?
14.
14.
14.
Very good. Okay, and if we range these 14, how we range them?
1 to 6 is
Mister,
acid.
1, 7, 14.
7 natural.
Yes, 1, 7,
Mister, 7
14.
is natural, 1 is acid, and from 8 until 14 space.
Very good, very good. So neutral is 7. Remember this one, neutral is 7, and at neutral you have same OH and H. While at the acid, what is more in the acid?
With the H plus.
H plus. Very good. The acids, they release H plus, and the base
OH.
OH, and there is an equal number of H and OH in the neutral. Yeah, okay, very good. Yes, very good. Remember that the gastric juice in your stomach are acidic because their pH is around 2 or 1 point something. So they are acidic, and many other are basic. Fine. What is a buffer?
Or mixtures that react with
Read
acids
the acid
or
and
bases or
basically
increase
all
the
pH with a specific range.
Very good, very good. So buffer is any solution that keep the pH within a specific range. For example, the pH of your blood is 7.4 just because of the buffers, okay, in your body. Fine. Next topic, 1.3, the building blocks of life. Any question till now, boys, before we move to next topic? If you
No.
don't understand anything in 1.1, like bonds and water and everything, and in 1.2 about the cells, that cells are the building blocks of life, we did it last 2 weeks. You can say it was very important topic, and we had discussed it a lot. First of all, carbon is like very important to all living things. Why? Because it is present in all molecules. It can make 4 covalent bonds, and it has like different shapes. It can make ring, chain, or branch shape, and it results in diversity of life. You can see in these pictures, these are not needed. You don't need to memorize them for the help to tell you. Okay, so no need to memorize these pictures. Now, what is the difference between a macromolecule and a polymer?
Macromolecules are large nucleus formed by joining smaller organic molecules together, also called polymers.
Yes, also they are called polymers. And what about the polymers?
Are molecules made from repeating units of identical or almost identical compounds called monomers, joined
Yes,
together by covalent bonds.
yes, very good. So remember that polymers are made up of many monomers, like many monomers join together to make polymer. This is the most important thing, that you say they have repeated units. Fine. We have 4 major macromolecules or the polymers to study. Please remember them. This table down will help us to just go over them quickly. We have carbohydrates, we have lipids, we have and we have nucleic acids. So these are the 4 major macromolecules. Now, please, very important is that learn their monomers. Like, what are their monomers? You must have to memorize them and then learn their structure. Here the structure is important. So carbohydrates, it is you can see that it is from carb and hydrate. So carb mean carbon, and hydrate mean H2O. So that's from here you can see that they have H2O, and then they have simple formula. If we say that their simple formula, so their simple formula is CH2O units. They are made up of CH2O units, like 1:2:1. So the carbon, hydrogen, and oxygen, I will write it here. 1:2:1 is the carbon hydrogen ratio over here. 1. Okay. What is their simple monomer? Either we call it as a monosaccharide or we call it as a sugar. So remember, their monomer is sugar or the monosaccharide. Next, lipids. their basic units are glycerol and fatty acids. So they are made up of glycerol and fatty acids. Proteins, they are made up of amino acids, and their main role is structural, not energy. You don't see listed energy here. You just see energy listed in the carbohydrates. You see energy listed over here in lipids, and also we have energy in nucleotide where you have ATP. So we have ATP, which is a strong energy molecule. Nucleic acids are made up of nucleotides. Okay, boys, again, what is the basic monomer of proteins? Yes, boys.
Ammonia acid.
Amino acid.
Amino acids, very good. So proteins are the polymers made up of many repeating units called amino acids. Lipids,
Monosaccharide.
Fatty acids.
the
Fatty acids.
monosaccharide is for the carbohydrate. Yes. The lipids, it is fatty acid and glycerol. Fatty acid and glycerol.
Glycerol.
Nucleic acid is made up of what?
Nucleotide.
Very good. Nucleotide. And the function of them is to store and transmit the genetic information. Now here, boys,
Like the
in this one by one we will go through, but if you understand this table, you can easily understand the whole, but because this table is bit blurred, I will go down. We discussed about this, the ratio. Okay, we discussed about this, the building block, this one, the monomer or the building block is the monosaccharide. Why not? Okay, let me take it like this. Okay, so remember that this is the building block. They can be monosaccharide, they can be disaccharide, and they can be polysaccharide depending on the number of the glucose join them. Then what is the structural and functional characteristic of lipids? We studied that lipids are hydrophobic.
Made mostly of carbon and hydrogen.
Yes, less oxygen, and they hydrophobic mean they can dissolve. They can or they cannot.
They cannot.
Yes, they cannot because they are non-polar. Now, three main types of them: triglyceride, phospholipid, and steroids. These are the three types of them. The difference between the triglyceride and phospholipid is mainly that tail. Why we call the fatty acids as hydrophobic?
non-polar because it's scared from water because
Yes.
it's hydrophobic.
Yes, very good. Non-polar because they are hydrophobic, and they hydrophobic mean they don't they are not attracted to the water. Okay, boys, this question is very important. The monomers of amino acids, what is the monomer of the protein? It is amino acid.
Amino.
Remember, and each amino acid has three parts.
Amino group, carbox group, a side chain called
Carbon group, hydrogen group, and the
R group.
Okay, very good. So we have an amino group, we have a carboxyl group, and we have a side chain. So remember that the only difference in each amino acid is what? The R group. Every single amino acid is different from others is because of the R group. Remember, so this is what makes the amino acid different. Then what are the functions? All these functions are written up in the table. Protein has many different structures like primary structure, secondary structure, tertiary structure, and quaternary structure. So you can see these in the folding here. And this is the structure of the amino acids. Always the R group is the variant. So please read these. Yani primary is a straight chain, secondary is folded, tertiary is 3D, and quaternary when many tertiary structures join together. Last one is nucleic acids. This is the monomer for the nucleic acid, and the monomer is nucleotide. Each nucleic acid has a monomer nucleotide, and these nucleotides has also three parts: the phosphate group, the sugar, and a nitrogenous base. There are 5 different nitrogenous bases. You don't need to memorize their name, but at least you should know that the DNA and RNA and ATP are the examples of the nucleotides. And ATP is a very rich energy molecule. This is also very important. A rich energy molecule is ATP. Okay, with this we reach to end of topic 1.3 also. Yes, boys, do you have any question till now?
No.
Thank you for the feedback. Anyone else, boys, do you have a question?
No,
Mr.
Next, cell discovery and theory. This is the second last topic, very easy topic. It states the 3 basic principles of cell theory. So who can say these quickly?
Mr. Bacterial cell,
All
animal
living
cell,
things
plant
are
cell.
all living things are composed, made of one or more cells.
Very
Cells
good. Continue.
Cells are the basic unit of structure and organization of all living organisms.
Very good. Read point number 3.
Cell arise only from previously existing cell, with cells passing copies of their genetic material on their daughter cells.
Yes, very good. So all cells divide and redivide to pass on the genetic material to the next cells, okay, the daughter cells. Okay, let us go quickly to difference between the prokaryotes and eukaryotes. We did many activities. Pro mean before, karyote mean nucleus. So before nucleus, they don't have nucleus, they don't have membrane bound organelles. They are very simple, but the eukaryotes are very complex. The example in the prokaryotes is bacteria. They are very small, but over here they are big, and the examples are the plants and animals. Boys, try to identify the cell. If it is given in the exam, like if this picture is given in the exam, they may ask you identify the cell in the picture. So you should be able to identify, because there is no mitochondria, there is no membrane organelle, there is no nucleus. So what it is? This one.
Mr. Bacterial cell.
Bacteria. And what is over here? What is this?
Bacteria.
Bacteria. So you see that there is no specific shape for the cell. They can give you any cell in the exam. Okay, now we have this labeling. This labeling is very important. You can do it. We did it many times. I'm not going to go into the details of it. Magnification eyepiece is always how much?
Times 10.
10x, yes. And the objective lens we have 4, 10, 40, and there is 100 also, but normally we don't use 100. So 4 is low. Okay, so we can say that like this: 4 is low power, then a medium power, and then the high power. Okay, so we have these 4 here. So please make sure that you learn them properly. Difference between the compound microscope and light microscope, electron microscope. The light microscope use light, and what is used by the electron microscope?
Use
Electrons.
Electrons. Maximum power of the
1000
compound
X.
1000, and here 500,000. Yes, it can observe
500,000.
living as well as non-living, the compound. But electron microscope,
only non-living things.
yes, very good. You can see colors in the compound, but you see only black and white in the electron microscope. There are,
Mister, what's
yes,
similar between it?
similar here.
Both provide the large images of something very small.
Yes, yes. What is the similarity between the 2 microscopes?
Both produce larger image.
Very good. Both help you magnify. Compound microscope don't have any type, but electron microscope has 2 types. So one is scanning, which is called as SEM. It gives you what?
3D images.
Very good. And the other one is a TEM, which is a transmission electron microscope. What does it do? It gives you details of the tissue. Okay, so it gives you tissue details. That's the most important thing you need to know. Any question from this topic, boys, before we reach to the last and then we go for the practice?
No.
No. Okay. What are the functions of the plasma membrane? Yes, boys.
The flexible boundary between the cell and its
Yes.
environment.
Very good.
Controls that enters and exits a cell like a gate around a school or
Very
home.
good.
It's selectively permeable.
Yes. So what? Yeah. What enters?
Cross.
What enters or what leaves in the cell? Now, quickly, the labeling. Like it's labeling. We have to label the parts over here, and it is easy to label in this picture. Let's say. So you have channel proteins which allow the materials to go in and out. You have three types of proteins mainly. You have inner proteins and you have these proteins. But more than important over here is this molecule. What is the name of this molecule?
What's called polar?
No, the whole molecule. The whole molecule.
Phospholipid layer.
Phospholipid. Yes, yes, phospholipid. And if there are if you call these two layers together, yani this is one
Layer.
bilayer. Very good.
Bilayer.
So yes, if you talk about these two together, you make it as phospholipid bilayer. Bi mean two, so it means that it is two layers together. Okay. Then we have a polar head. We spoke about it already. Hydrophilic. Then we have a hydrophobic, which are the fatty tails. And this is the same explanation again and again. What is the function of these, boys? This table is very important. I'm writing it here, like, because it has I will write it here because this table has the functions. Okay, so this is very important. Don't miss anything from here. So what does it says? It says that what are the functions of the cholesterol? So cholesterol means
The fatty acid and
Fatty and acid chains for
the
the
sticky
lipids.
mixture is sticky together.
it means that it increase, I will say here, it increase the strength and fluidity of the cell membrane. It's because of the cholesterol. Okay, fine. Carbohydrates, they are very important for signal and cell recognition. So two things. Okay, then proteins. How many types of proteins we have? Three. Very good. Remember them, boys. Three types. Yeah.
Proteins.
The outer surface and
Yes.
the inner surface.
And throughout, yani start from one side and go to the other side. Okay, so these are the other proteins. Last one, boys, fluid mosaic model. A fluid mosaic model says that it is a sea of floating moving phospholipids, cholesterol and protein. It means that everything over here is moving. Okay, so that's what we say, that it is a floating sorry, like this. Okay, and the last one again, boys, it's a difference between the prokaryote plants, animals and bacteria. So plants and animals are eukaryotes.
This one also very important.
Yeah, definitely, it's coming again and again. Chloroplast is in the plant, cell wall is in the plant, no nucleus, but it has cell wall. I will write it here down. The bacteria has cell wall. So this is common between the plant and bacteria. They all have DNA. Yeah, I will write down here. Look, all have plasma membrane. What else? DNA. What else?
RNA.
Cytoplasm. And the last one was what? Ribosomes. Thank you so much, boys. Ribosomes. Yes. So they all have these four together. Okay. With this, we reach to end of the whole revision from the summary, boys.
What is the better, we study from the PowerPoints or the course specifications?
Boys, I will recommend you to, they are all almost same with little plus minus, okay? But I will recommend you to study from the course specification and Al Diwan book.